All right, so good evening, everyone. Hello. If you can hear me, kindly drop. If you can hear me, kindly drop an emoji. Good evening. Okay, Fati Warrioluwa, Adekola, Ademola. All right, you are welcome. Others will join us as we continue. So, how was today?
Fine, ma'am.
Yeah. Um. Yesterday when we met, we I spoke on some tips on answering questions, and I promised to come back with some diagrams today. So I will attend to that. Then yesterday as well, we looked at three out of the specimens that you would be working with. I want to ask, did anybody practice anything at home? Probably the
Yes, ma'am.
diagram. Okay. Which one did you practice?
The water leaf.
You practiced the water leaf. All right. That's fine. And how well were you able to draw it?
I'll just rate myself 6/10 since as
Six
I
of
was doing it, yes, I was still looking at somewhere before I can draw it, and I still need to learn how to draw it off it.
offhand.
Yes, ma'am.
Okay. And was the diagram neat? Was it rough?
Yes, ma'am. It was neat.
Neat. Okay. Okay. That's good. Ademola and Ajayi, I think you're also part of us.
Yes, ma'am.
All right. So did you join yesterday's class? Ademola, did you join the class yesterday?
Yes, ma'am.
Yeah. And did you practice any of the diagrams from the specimens we treated?
No, ma'am.
Why? Were you having paper today?
No, ma'am.
So the best way to it is to practice. That's
Okay.
something about biology. You just have to practice. You just have to practice and practice it, at least for the sake of this exam. Okay.
Yes, ma'am.
Ajayi, are you with us? I'm not sure. So let's get started. And I'm going to start from before we continue with the specimen. I want to revisit. I want to revisit the do's and the don'ts when you are doing your practical and when you are drawing your do's and don'ts. So I'll be sharing my screen now. Okay. So number one thing is that whichever diagram you are asked to draw, you know, the questions we are treating, they are just speculations. Say, saying maybe they might ask you this, they might ask this from you, they might ask this from you. So whichever one you were asked to draw, ensure you draw what you see. Ensure you draw what you see. And then, like I mentioned yesterday, whenever you want to draw, make use of long pencil, at least 15 centimeters long. It helps your hands, it helps your hands to move freely on your paper, and ensure that you are not impressing much force such that when you have to clean, the impression will still be there. Okay, for your papers where you want to draw, They would have created the space. There is no room for, okay, this diagram is not neat, I'm going to the other side to draw. No. So manage that space you were given as much as possible. Then don't draw using cut lines. Don't draw using sketches, as if you are working on hearts. I will show us some images. Don't sketch, don't sketch, don't sketch. Then make your diagram large enough to the exact size as required. Waec will always come with size, either 6 to 8 centimeters, 10 to 12 centimeters, 8 to 10 centimeters, or whichever. You will be given size. So, and I will also show you how to do that. Then when you want to draw, ensure that you keep space to your right hand side, so that when you want to label, you can label there. Something like this. You have the space to your right hand side to label. Do you understand? So don't exhaust all the space. Ensure that you have space to your right hand side to label. Then if you want to label, make use of your ruler. Hello, your ruler is not for fashion parade. You must use it. You can see these lines, they were all drawn using ruler. Make use of your ruler, please. Make use of your ruler. Don't use hand. Then whatever you want to label, ensure that your line is touching that part. Look at the seed that was labeled here. The seed that was labeled here, excuse me. My line touched the seed. Exactly. Can you see it? So if you also want to label, your line must touch what you are labeling. Then don't use arrow. I did not use arrow here, although in some images that I would use, they used arrow. It is incorrect. I tried to source for images that didn't have arrow, but have the information that I needed, but I couldn't get. And that was why I picked those with arrows. But what is expected of you is that you don't use arrow. Hello, don't use arrow to point. Just draw your line and make sure your line is touching the parts you are referring to. I hope we are following. Then all your
Yes.
lines must be parallel to each other. Can you see this? There is no line crossing each other. Look at this one. Look at this one making use of arrow. It is wrong. This other one too, crossing each other. It is wrong. Your lines must not cross each other. You must not make use of arrow at the same time. Do you get it?
Yes, ma'am.
All right. So then your labels, they must align. Okay, I think you can still see it faintly. If your pencil is very faint, you can, if your pencil is very faint, you can draw a straight line using your ruler down like this. So this will be a guide for you, telling you that all lines that is coming from your diagram must stop at this line, and your labelings will start immediately after that line. Then when you are through, you don't leave the line there. You clean it up, you erase it. Okay, you clean it up. You clean it up So that is why when you are drawing, you make the line very faint, very faint. Do you understand?
Yes, ma'am.
Yeah.
Yes, ma'am.
Then do not shade. Mentioned that yesterday, and that was why I took my time to draw this diagram and intentionally made some errors and I marked it. Do not shade. Do not shade. Um, go away. If you look at the side, you see that it is a shaded part. I also wanted to indicate seed here. I shaded it. It is wrong. You don't do that. If you want to show us your seed, just like I did here, is sufficient. Okay? Do not shade. Then don't make mistakes like this. What is here? You can see lines crossing each other. Lines crossing each other. It is very wrong. By the time your examiner wants to mark, this is what your examiner will be looking out for. Once there is a mistake, your examiner will circle it. Another mistake, your examiner will circle it. By the time your examiner circles about 5 to 10, you will lose a particular mark of about 2 to 3 marks. I hope you are with me.
Yes, ma'am.
Yes.
So you must be patient. Ẹ jẹ́ kára yín balẹ̀. Don't just be patient. Be calm as much as you can to have a perfect diagram. I always emphasize on this because if you have a diagram of 12 marks and you manage to get 10 marks, of course you have done wonderfully well. Do you get it? Um So do not shade. Peradventure you need to indicate anything. You can use dotted line or straight lines. These ones, I'm using it, these dotted lines, I use it to show that, okay, oh, this part is a succulent part, so it is filled with water. It has tissues that is filled up with water. Instead of me to shade like this or to do something like this, like this, like this, like this, it is very, very wrong. Please don't commit such blunder. All right?
Yes, ma'am.
Um, then when you have a cut surface, a dissected surface, when you have a dissected surface, you or thickness, you will indicate it using two lines. That is why you have this orange like this. Any diagram I see and I see two lines, double lines like this, it is telling me that this is the dissected surface. It is telling me that this is the dissected surface. So peradventure you were asked to draw any, for example, the egg would be dissected, the boiled egg would be dissected, the orange would be dissected, the pineapple would be dissected as well. I don't know if you will eventually dissect the water leaf. Probably you cut the stem to look at the vascular bundles or something. Once it is a cut surface, the external lines are represented with double lines. Do you get it? And when you are doing it, you make your hands to be straight as much as you can and you don't make it to overlap. Can you see that? It must not overlap. Are you with me?
Yes, ma'am.
All right. Compare this one that I drew and compare it with this. Can you see? Something is already wrong there, and that is what the examiner would be looking out for. All right. So then you must not forget your title. Title is cut separately. Title is either 1.5 mark or 2 marks. Your title. And your title is always below your diagram. See the title here. Your title is always below your diagram. Don't put it above. It is wrong. Once you put it above, there is no mark. No mark for that. You put it below. Are you with me? So don't forget to put your title. And please, pen must not show in all these things we are doing. Your diagram, your labelings, your title, your magnification. Don't use pen. I want to use pen to decorate and beautify. No, 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 no. You would be penalized for doing such. All right? Then magnification. Magnification of your object is very, very important. So, and this is where the function of your ruler comes in. And how do you calculate your magnification? This is the formula for calculating magnification. You use the length of the specimen you have drawn. So it is the ratio of the specimen you have drawn to the live specimen you were given. The size of the specimen you have drawn to the specimen you were given. Are you with me? Now, so let's assume that this is a cut surface and I was told to draw 0 to 8 centimeters. So this is about almost 8 centimeters long. Let me approximate it at 8. Hello?
Yes, we can hear you, ma.
All right. So let's assume that what I have drawn is 8 centimeters. Using ruler, you will read from here. Excuse me. You will read from here to this point, okay? You will measure it. Let's assume it is 8 centimeters. 8. You will now use that your same ruler. The specimen you were given, you will measure the cut surface as well. It could be small, it could be bigger, okay? So let's say when you measured your specimen, it's about 12 centimeters or 14 centimeters. So you put that value here, all right? And you will use your calculator to do the division. 2 here 4, 2 here 7. 4 divided by 7, mathematician, that is 0.7 in 40. 5, 5, 6, 9, 7 times 5 times 5. So it's around 0.5. Okay? 0.5 something something. Are you there?
Yes, ma'am.
So what you will do after you have calculated your magnification, this calculation must not show in your booklet. You do your calculation somewhere else, okay? You just measure it, press it on your calculator somewhere, and you come under your diagram. You will put magnification is equals to times whatever you have gotten. Excuse me. Are you there?
Yes, ma'am.
The magnification is equals to times whatever you have gotten. Note, magnification does not have units. It is just telling us that, okay, what I do, it is half the size of the real specimen that was given. It is half the size of the, it is times 2 of the specimen that I was given, okay? And so I brought this here. This is Waec reports. Can you see? Quality,
Yes.
this is how it would be marked. Are you the only one there? I hope others are following me.
Yes, ma'am.
All
Yes,
right.
ma'am.
Yes, ma'am.
Your neat labels will be marked. Your lines, how clear are they? It is going to be marked. Your size, if it is correct, it is going to be marked. Your magnification, it is going to be marked. Your cut surface, did you represent it with double lines? That is, if there is double line, it is going to be marked. And all these are details. Did you show the placenta? Did you show the succulent ear? Labels, did you show us the epicarp? Did you show us the mesocarp? Did you show us the endocarp? Did you show us the placenta? Okay, so that is how those the marking is being done. Then getting your size too. How do you do that? Let's assume this is your plain sheet and you were told to make a diagram of 8 to 10 centimeter long. Are you there?
Yes, ma'am.
All right, 8 to 10 centimeter long. What you will do is you will pick your ruler and you will place it in this direction. Okay, on your paper you will use dot to mark point 0 from your ruler. Okay, you will mark this point 8 and you will mark point 10. If it is 6 to 8 centimeters, you will mark your point 0, your point 6, your point 8. Do you get it? So let me work with this. Let's assume that we are using 6 to 8 centimeters. So what that means is that your diagram must not be smaller than 6 centimeters. What it means is that your diagram must not be smaller than 6 centimeters and it must not be longer than 8 centimeters. Are you with me? Any diagram below 6 centimeters will be penalized and any diagram beyond 8 centimeters will not be marked. As in the mark for the size, you won't get it. The candidate won't get it. So you pick your ruler, you mark point 0, point 6 and point 8. So your diagram will be within this range. Do you understand?
Yes, ma'am.
And so that is how to take your measurements. Look at this one that I did. It must not have lines like this. I just made it to have line for explanation's sake. But you can see that it was dot initially, point 0, point 6 and point 8 centimeters. So that will be your guide for the size of your specimens. Do you understand?
Yes, ma'am.
All right. So that is that about that. Now let's go back to our specimens that we are treating. We've treated specimen A, specimen B, and specimen C. So let's move on to specimen D. Specimen D is a quill feather of a domestic fowl. Specimen D is a quill feather of a domestic fowl. So if you are asked, identify specimen D, your answer should be quill feather of a domestic fowl. All right? Quill feather. And your spelling is very, very important. It must be correct. Your letter I must not look like L. I said that yesterday. Okay? So you could also be asked that what are the functions of the feathers? Functions of the feathers is that used for flying. It is used for flight. It's still the same thing. Okay? Are we there?
Yes, ma'am.
All right. Thank you for following and responding, Nore. Others to make sure you are following. Then
Yes,
it is
ma'am.
also important for insulation. In other words, it regulates body temperature. It regulates body temperature. Then also remember, we are talking about these birds are found in group of animals that are called aves. Gosh, I want to annotate too. All right. It is found in group of animals that are called aves. Aves. These are birds, and they are homeothermic animals. Homeothermic animals meaning that their body temperature does not change with the temperature of the environment. Their body temperature remains constant and unchanged. All right. What are the adaptation of the feathers of that particular feather, quill feather? Number one is that it is very light in weight. It is very light in weight. So it makes flying easier for the bird without adding extra weight. Okay. Then it also has bubbles and hooves interlock. It also has bubbles and hooves interlock, which makes it easier for the bird to move in air. That is aerodynamic ability for the bird to move in air freely. Then the main parts of the quill feather, we have the calamus, we have the rachis, we have the barbs, we have the barbels. Barbs and barbels. Barbels are down there. I tried to get an image that has all those parts, but then I still want to get an image that can easily be drawn by students. Okay. So the calamus, that is the hollow basal part inserted into the skin. So this part down here, that is the calamus. This part down here that is inserted into the skin is the calamus. Okay. We also have the rachis, which is the central shaft. That one is above the calamus, rachis around here. Instead of having the quill, we also have the rachis. Okay. We have the barbs, which are the branches, and we also have the smaller branches, which are called the barbels. So these are the barbs. Can you see it? The barbs. We have the shaft, which looks like the midrib, and we have the vein above the barbs. Okay.
Ma, excuse me, ma.
Yeah.
What's the difference between the vein and the barbs?
This particular author labeled the barbs as the vein. Okay, it is the barbs that forms the vein.
Okay.
It
Okay.
is the barbs that forms the vein. All right.
Okay, ma.
So aside the quill feather in birds, we have other types of feathers. We have the contour feathers. We have the down feathers. Okay, you could be asked that other types of feathers that could be found in the animals that possess that. We have the contour feathers, we have the down feathers. Then the feathers, what makes up this feather? It is keratin. Feathers are made of keratin. They are tough. Keratin makes the structure tough, even on the human skin, in the mammalian skin too. We have the keratin, which makes the surface of the skin tough, okay, and flexible. So the feather is made of keratin. Um, a question could come relating this to scale, asking that why is the quill feather considered a modified scale? And that is because it evolved from the reptilian scales. You know, this is making reference to evolution, making reference to evolution. And scales also, they are composed of keratin as well. So the feathers are also composed of keratin. Keratin. Other feathers and their functions. I've mentioned two above: the contour feathers, the down feathers. We also have piloplumo feathers. Okay, that is another type of feathers that can be found on birds. You know, we are just having a broad scope of where questions could come from. Now, if you look at quill feather and water leaf, their structure looks somewhat similar. So a question could come asking if there is any similarity or differences between the two. Of course, there are differences, so many differences, okay? But for similarities, I'm not sure there is any. I'm not sure there is any, but then there are similarities. There are differences rather. Now, if you are asked to differentiate in tabular form, make your table a table. Hello? Make your table a table. Draw a real table. Draw a real table. Put your water leaf, specimen, whatever. Put your quill feather, specimen, whatever. Don't just do like this and begin to write number one, number two. No. And when you are differentiating, your differences must correspond. So let's look at the differences between water leaf and the quill feather. Water leaf is a plant, true or false?
True, bro.
Hello? Yeah.
True.
And quill feather is part of a bird. So it is from animal, okay? Water leaf, they are used for making food. The leaves carry out photosynthesis. But for quill feather, it is for flight. It is a part of the plant that is used for flight. Water leaf is made of cells and water, but quill feather is made of a tough protein called keratin. Water leaf growth is continuous, non-stop, but quill feather, they have a fixed size, and at a point they can fall off. You know, growth in plants is unlimited, while growth in animals is limited. Water leaf has green color, has green leaf, but quill feather, they just have hard central shaft with soft barbs. Okay, they are not green. They are not green. All right. Um, that is the differences between water leaf and quill feather. Let's move on to the pineapple fruit. So I include this image in order to create a sense of easy diagram, because in your heart of hearts you may look at it, hey, this pineapple that is dissected, how can I draw it if they asked us to draw it? This can serve as a template for you. Are you with me?
Yes, ma'am.
Yes, and then you include other parts. You can include the stalk. Can you see that it has started looking like a um a pineapple? Then you partition it, you partition it, you partition it, you show us the seed. Okay, you can go ahead and include eyelets to the side, because the outer part of pineapple is not very smooth. And when you are done, you can have these labelings. So the tip here is the crown. The tip is the crown. We have the outer part, which is the epicap. In between, we have the ovary. The center is called the axis. The axis. And here we have the stalk, the plant stalk, or the fruit base. Okay, so let's briefly look at the pineapple. And the first question is, you can also be asked to identify it. It is a pineapple fruit. All right, pineapple fruit. So you could be asked that what type of placentation is found in this specimen, and the placentation is parietal placentation. Parietal placentation. Okay. Economic uses of pineapple. Pineapple is a fruit, fresh fruit. It is rich in vitamin C. It can serve as raw materials for fruit and juice industries. Okay. It can also be used to make fruit salad. It can be processed into canned fruit. It can be processed into wine. It can be used in baking when making fruit cakes and the likes. It can be baked. Okay. So these are some of the economic uses of pineapple. And pineapple is a type of fruit that is formed from multiple fruits, also called composite fruits. Reason for that is because it is formed from group of flowers that are fused together. That is why we have islets. That is why the so that is I'm coming. I want to annotate. I don't know. So that is why, okay, that is why the pineapple has eyelets. It has eyelets because it is formed from many fruits fused together. Okay. Eyelets. Each what happened now? Okay. Each eyes you are looking at is an eyelet, and it is representing a flower each. So it is many ovaries from many fruit flowers that fuse together to form that. All right. What
Right.
is the edible part of the pineapple fruit? The fleshy part is the edible part. Same thing, the orange. The fleshy part is the edible part. Okay. And how can pineapple be propagated? Pineapple can be propagated by planting this crown. You will remove this crown, this shuku edge, and plant it. That is how pineapple can be propagated, and it is an example of vegetative propagation. So you can put planting of planting of crown, yes, or you call it sucker, and you put it in brackets, vegetative propagation. Do you get it?
Yes, ma'am.
Yeah, pineapple is also considered to be a
Yes,
fleshy fruit. Pineapple is also considered to be a fleshy fruit because it is juicy and it has soft tissue. It has soft tissue. What type of inflorescence gives rise to pineapple fruit? A spike inflorescence. The cluster of the flowers formed spike, and all these are grouped together fruits. What are the adaptive features? What are the adaptive features? Of course, the pineapple fruit has spiny leaves. It has spiny leaves or the crown, and the spiny leaves, note that spiny leaves is a modification, or that same spiny leaves is also the crown is thick and it is waxy. They are all these are modifications to reduce loss of water from the plant. Okay, they are all modifications to reduce loss of water. Another modification is that it has crown and sucker for propagation. It has fleshy and juicy mesocarp. It has fleshy and juicy mesocarp. It has multiple fruit structure of fused fruitlets. The epicarp is tough and rough at the same time. It is tough and rough at the same time, and it is a modification to also reduce water loss. It is a modification to reduce loss of water. I hope we are following.
Yes, ma'am.
All
Ma'am, can you
right.
also say fleshy and juicy interior as written?
The mesocarp, the endocarp,
Ma'am,
rather. Yeah.
it's written interior. The place you said it has fleshy and juicy interior. Can
Interior,
we also
yes. You okay? Endocarp. Thank you. So that's that about that. All right. Um, let's look at specimen F, and that is where we will call it a day for today. Specimen F. And our specimen F is what?
orange?
Orange. Orange fruit. Orange fruit. Transverse section of orange fruit. This is the diagram showing orange fruits. We can see the epicarp, which is the outer layer, the mesocarp, the endocarp, which is the fleshy and juicy part, the seed, which is used for propagation. The placenta, which shows the arrangement of seeds, okay, the point of attachment. Then the succulent ear, then you can see the title it follows immediately. Don't forget, if you are asked to draw, your title must follow. So if you are to identify the fruit, it is what? Orange fruit. Although orange fruit is a fleshy fruit and it is also a berry, a type of berry. The fruit has 3 observable layers. It has the epicap, which is the outer part, the peel. It has the mesocap, that is the part between the epicap and the endocap, which is the white spongy middle layer. It also has the endocap, which is juicy. Okay, it is juicy, and that forms the inner segment that holds the juice sac. What is the type of placentation that is found in orange? Axial placentation. So that forms a difference between the pineapple and the orange. Orange has axial placentation. Pineapple has parietal placentation. Okay. Then the endocap is the edible part of the orange. The endocap is the edible part of the orange. How can orange be propagated? Orange can be propagated both sexually and asexually. Sexually using seed, and vegetatively by grafting and budding. By grafting and budding. Vegetative propagation. Okay. Orange is also a simple fruit. It is a simple fruit, but it is formed from ovary of a single flower. It is formed from ovary of a single flower. Economic uses of orange. Yes, economic uses of orange. Orange can be consumed fresh. That is what we use it for majorly. Orange is a source of vitamin C. Orange is a raw materials for juice production. It is used as flavoring for food, for drinks, and baked goods. Are we together?
Yes.
The peel is used for essential oils. In cosmetics, the peels is used for cosmetics. It can also be used to make jam and jelly. It serves as animal feed as well. So it serves as source of employment for people that have orange plantation. That is another one. So for farmers, orange serves as source of income. Those are the economic importance of orange. Economic importance of orange. Examples of other fruits that have similar placentation with orange. Garden egg has similar placentation, axial placentation. Pepper has axial placentation. Tomato has axial placentation. Watermelon has axial placentation. Okay, so this group of fruits has the same placentation with orange. Are you there?
Yes, ma. Yes, ma.
Yes.
Yes. So let's look at similarities between pineapple and orange. We need to compare the two because they are both fruits. They are both fruits. So both of them are fruits. That is one similarity. Both of them are fruits. They are grown for their edible and juicy fruits. Both are fruits. Another thing is that they are both fleshy fruits. They are both fruit crop. They are both fleshy fruits because they have succulent parts. All right. Both can be propagated vegetatively. You can use sucker for pineapple and you can graft orange. Do you get it? Both contains vitamin C and both are raw materials for juice industry. That is their similarity. But then, let's take it a step further. Most times the question is always observable similarities, meaning what can you see as you are looking at the two fruits? What do they have in common? Are you there?
Yes, ma'am.
Yes, ma'am.
So one of the observable similarity, if you hold pineapple on the right hand and you hold orange on the right hand, you will see that both are fleshy fruits. Both are fruits. Then both have brightly skin color. When they are ripe, they have like yellow to brown outer skin color. Okay. That is their epicarp. Their epicarp has bright color. Then they contain multiple seeds. In oranges, you can see the seed. In pineapple, you will see the traces. O's, you will see traces of seeds. Then they both have segmented inner structure. Look at the orange. This is segmented structure. Okay, that is for the orange. And for the pineapple, you can see the segmented structures too. You can see the segmented structures. Okay, so they both have segmented structures. Then they both have high water content. That is why they are juicy. They both have high water content. Let's look at the observable differences. Observable differences. Number one, we can talk about their placentation. Pineapple has parietal placentation. Orange has axial placentation. Okay. Then when you look at their epicarp, for pineapple, the epicarp is rough and scaly. That is observable feature. The epicarp is rough and scaly, but for orange, it has smooth and leathery epicarp. Okay. Then if you go beyond observable features, you can also say that pineapple can be propagated by sucker, while orange can be propagated by seed. Pineapple is a multiple fruit, while orange is a simple fruit. Okay. Then pineapple is a biennial crop. It is a crop that it takes it 2 years to complete its life cycle. So if you are to classify both plants according to their life cycle, pineapple is a biennial crop, orange is a perennial crop. Perennial crop are otherwise called permanent crop. It takes them more than 3 years to complete their life cycle. Do you get it?
Yes, ma'am.
So if you classify them based on their life cycle, pineapple is biennial, while orange is perennial. Okay, so we are going to stop there for today. um Tomorrow we will look at the egg. Of course, I've meshed this together, but it's my partner that is going to take you that most likely. Okay. um for your fresh egg, you are going to carry out protein test on your fresh egg using Biret tests. You will use sodium hydroxide and copper sulfate on albumin. Albumin is the proteinous part of the egg. So my colleague will facilitate you on that. I don't know if you have any question on this that we have done this evening.
I just want to know if the magnification you said you would teach us, if it has been taken.
Yes, it has been taken. So it means you joined late. So I will quickly go back there. Magnification. If you want to, once you are drawing your diagram, were you there when I take the size?
No.
Wow. So I'll quickly rush through this again. So if you would be given a live specimen and you'll be asked to draw. Now, you could be asked to draw a diagram that is 6 to 8 centimeter long. Okay. So on the space provided for you, make sure you manage that space very well. Pick your ruler. Your ruler will stand like this. Your 0 point will start here, 1, 2, and it goes down. So on your ruler, you will mark point 0, which is the starting point. Are you with me?
Yes, ma'am, I am.
You will mark point 6 and you will mark point 8. Now, if you are asked to draw between 6 to 8 centimeters, you are only being told that whatever you want to draw should not be smaller than 6 centimeters and it must not be bigger than 8 centimeters. Okay? It may not be 8, it may not be 6 to 8, it could be 8 to 10 centimeters. What it means is that your diagram must not be smaller than 8 centimeters and it must not be bigger than 10 centimeters. Okay? So you will pick your ruler the same way. You will mark point 0. Which other points will you mark for this one? Point 8, which is the minimum, and point 10. Do you get it?
Yes, I do.
Do you understand what I'm explaining?
Yes, yes, ma'am.
Yeah. And so within this range, you draw whatever you want to draw. You will ensure that it doesn't go beyond this mark and it doesn't go, whatever you have drawn should not be below this point. Once you are done drawing, you calculate your magnification. How do you calculate magnification? This is the formula for calculating magnification. The size of what you have drawn over the size of the specimen you were given. You could be given, it could be pineapple you want to draw. Your pineapple could be 30 centimeter long. Okay, so let's assume it is pineapple you drew and it is 30 centimeter long. What you drew is 30 centimeter long. What you were given is 30 centimeter long and what you drew is 10 centimeters. By the time you do your division, you are going to have something like 0.3. Am I correct?
Yes, ma'am.
Yeah. So your magnification will be written as magnification equals to times 0.3. It will not have any units. So to calculate your magnification, you will put the size of what you have drawn on top, and you will put the size of the specimen you were given below. Do you get it?
Yes, ma, I do.
So please, for your centers, listen to your biology teacher, because he's the one that will give reports. If you were given lime as orange, that is what he will report, and it is the magnification they will use to mark you. But make sure that after you are through drawing, after you are done drawing, you put your magnification and you also put your title. Let your magnification be below your title. Don't forget your title, don't forget your magnification. Do you get it?
Yes, ma.
Do you understand it now?
Yes, I do.
All right. And so that is how to calculate your magnification. Any other question again?
Since we are giving either 8 to 10, can we draw 9?
If you are taking 8 to 10, yes, you can draw 9. It must not be smaller than 8. You must not draw 7.9. You can draw 8. You can draw 8.1. But you must not draw 10.1. Do you get it? Do you get it?
Yes, ma, I do.
So, yeah, you will draw according to the question you were given. The question will state it. Draw specimen A, so centimeter to so centimeter long. So it must not be smaller than the smallest centimeter, and it must not be bigger than the biggest centimeters. Do you get it?
Yes, ma.
All right.
What
I
if
don't know
the
if you
space measure the
okay.
ruler? Like we being weevil cannot be measured with ruler. So how can we measure the such
What is it that
specimen?
you cannot measure with ruler?
Being weevil
Bean weevil.
is smaller.
Bean weevil. Okay. So for that, it is an estimation. So let's assume you draw something about 6 to 8 centimeters for your diagram, and your bean weevil is about 2 mm long. 2 mm long. So you are going to have something like times 10 times 15. Do you get it?
Okay, ma.
Do you understand it now? Yeah. Any other question? Any other question? Okay. So in the absence of none, let's stop here for today, and we shall continue tomorrow again. Okay?
Okay, ma. Thank
All
you.
right. Thank you all for your patience. Thank you for attending the classes. I wish you all. Ma, please take us on the remaining one. Hello?
Hi.
Somebody said I should take you on the remaining one. We have shared the responsibilities. The best I can just do now is to rush through it, but already I'm 10 minutes behind schedule. Wouldn't you mind if I rush you in the next 10 minutes? Esther, you spoke, oh, you are not answering.
I'm here, ma. You can take
Hello?
us, ma. I don't mean today, ma.
I will rush you through it, but okay. Hello, what is the house saying?
You can go, ma.
Ore, Agu, Ademola, Elizabeth.
Yes, ma.
One is low,
You can
but
go.
since everyone wants us to go on, we can go on.
It's not everyone, it's just one person that spoke.
Yeah, ma. You can go on.
I won't take, I will just rush through it, and then I won't drop the material today as well. Probably by Saturday, that is when I will drop the remaining material. But the one I today, that is the pineapple feather. You will get today, okay? Over time, let me quickly rush through this. Yeah, so for the remaining specimens, we have eggs, we have fresh egg, we have boiled egg, and we have water. So for the I'm presuming that they might ask you to carry out protein test. Okay? I'm might ask you to carry out protein test. Are you there? And when you
Yes,
want to
we are.
carry out the protein test, the part of the egg that is proteinous is the albumin, that is the egg white. That is the albumin. That is the portion that is proteinous, and that is the part you are going to use for the test. You will just put a little in the test tube, add your reagents. I'm coming. Where is it? I'm trying to look for it. Add your reagents, the sodium hydroxide and the copper sulfate. When you do that, if you have done it before, that process is called burette test. Please note. That process is called burette test, and it is a protein test. So when you do that, you will observe purple color or violet color. That would be a color change in the test tube. You either have a purple color or a violet color, okay? And once you have this color, it is an indication telling you it is a positive test, telling you that protein is present. So your conclusion is the food substance contains protein, or protein is present. Do you understand? But the
Yes,
name
ma'am.
of that procedure is burette test. Burette test. So briefly, let's talk about the egg itself. The egg itself, it's a gamete. A gamete for reproduction. Okay? If you dissect the boiled egg, this is what it's going to look like. If you boil the egg and you dissect it, you have the inner part, which is the yolk. The yolk has the germinal disc, which is the point of germination. We have the albumin. We have the chalaza. We have the shell. We also have the membranes. I'm not sure. Okay, you can see we have it should be around 3 membranes. Yes, we have the outer shell membrane. We have the inner shell membrane. We also have the vitelline membrane. That is the yolk membrane. We have the ear sac. These are the parts of a boiled egg when you dissect it. Please, when you are labeling, don't use arrow. The use of arrow here is very, very wrong. Okay? So the external part of the egg, we have the shell, we have the shell membrane, we have the ear space. And when you break it, okay, the ear space, that is when it cracks. Internally, you have the egg albumin, also known as the egg white. We have the yolk, we have the chalaza, we have the germinal disc, we have the vitelline membrane. Now, what is the function of the eggshell? Eggshell protects the content, the inner contents of the egg, number one. Then it is permeable. What does it permit? It permits exchange of gases. It permits oxygen to go in, and it also prevents water loss to an extent. Okay. It also forms barrier. It also forms barrier against microorganisms. And for organism that produces this egg, they carry out sexual reproduction. So birds are oviparous in nature. What does it mean? It means they lay their eggs and then the embryo develop outside the body of the female animal. That is what we call oviparity. Are we following?
Yes, ma'am.
Yeah. So the albumin, which is the egg white, what is the function? That is what supplies the developing embryo with water and protein needed for growth and development. The developing embryo is supplied with water and protein by the albumin. That is why the egg albumin is the proteinous part of the egg. The egg yolk is not proteinous. Okay? But the yolk is actually the main source of the food for developing embryo. In all eggs, the yolk is rich in nutrients for developing embryo. While the chalaza holds the yolk in place. Look at the image. You see the chalaza to the upper parts, to both sides, to the right and to the left side of the egg. You can see the chalaza. So that is what is holding the yolk in position. It is holding the yolk in position. And the air space helps in exchange of gases for the embryo. Okay. As the embryo grows, the airspace increases. As the embryo grows, the airspace increases. Now, the egg, if it is, like I told you, it is a gamete. It is a gamete. So when it becomes fertilized, it becomes what? A zygote. It becomes a zygote. Then the type of development that is found in animals that possess that egg is external development. External development. The shell is a bit tough and hard, and it is because it contains calcium. So it is mainly made up of calcium carbonate. The shell is composed of calcium carbonate. Egg is a complete food, and it is because the shell consists of calcium, the albumin consists of protein, the yolk consists of other nutrients such as fats, the vitamins, and essential minerals that are needed for growth and for development. Okay. For the germinal disc, it is important in fertilized egg because that is where fertilization will occur. That is what we fuse with the germinal disc is like the nucleus of that egg. That is what we fused with the nucleus of the sperm. Okay. And so that is the point at which the embryo will begin to develop. That is where the embryo will begin to develop from. Okay. So I've taken this test for you. Now we have water. I've been thinking, I've been cracking my brain. What are the likely questions that could be asked? Of course, I couldn't get. But what came to my mind is the importance of water to plants, importance of water to animals, importance of water to the ecosystem. Okay. So, and also importance of water in reproduction. For reproduction, water transports gametes and also helps in the movement of gametes. For example, the pollen grains in plants. Okay. It also serves as medium of fertilization. For example, amphibians and fishes that reproduce in aquatic environments. Fishes and amphibians that reproduce in aquatic environments, they need water for fertilization to take place. It also hydrates the reproductive cells. Water hydrates the egg cells, it hydrates the sperm cells and keeps them alive. Okay, the developing embryo also needs water to grow. Seeds that want to germinate needs water to grow. So water is essential for seed germination. It is essential for development. Importance of water to plants. Water is needed for photosynthesis. Water is needed for transportation of nutrients. Water is needed to maintain turgidity, especially in herbaceous plants, such as the water leaf you will be working with. Water is needed for turgidity. It also controls temperature. It cools the temperature of plants and animals. Okay, in plants it cools the temperature through transpiration. That is the removal of water in form of water vapor. In plants also, water is needed for germination. Okay, it is needed for germination of seeds. And in animals, water is needed for digestion, water is needed for excretion, water is needed for hydration, it is needed for circulation, water is needed for regulation of body temperature. All right. In the ecosystem, water supports all lives. Water supports lives of plants, lives of animals, lives of terrestrial organisms, lives of aquatic organisms, lives of arboreal organisms. All lives depends on water. Okay? Water forms habitat for aquatic animals such as lakes, the rivers, the oceans. It also controls Earth's temperature. Water moves nutrients around through rain, rivers, and soil. Water is needed for pollination. Water is also needed for seed dispersal. Water is needed for pollination. Water is needed for seed dispersal. So those are the importance of water to man, to plants, to animals in the ecosystem, as well as reproduction. So I think I have to stop here. Then if you are asked to draw egg also, remember it is a cut surface. This yellow line, it is a thick surface. You must represent it with double line. Do you get me?
Yes, ma'am.
All right. I don't know if you have any question.
Ma'am, I just want to get settled the last question. Assuming the specimen A is 1 inches. So how did you say I can measure it?
I didn't get that question. Can you please come again?
I want to settle on the last question I had. Assuming specimen A, which is too small to measure regular. If
It's too
assume is
small.
one
Well,
millimeter.
I'm not hoping that they will say you should draw it, but peradventure it happens. Peradventure it happen. Your magnification will be a large number, like times 15, times 20, so to say, depending on the size you were given. Okay?
Okay, ma'am.
Yeah. So you will also have to check with your biology teacher in the lab as well. Of course, your biology teacher, it is part of it. Your biology teacher will be with you throughout the practical. So if you have any question, you can always beckon. Okay? But I'm not too sure they can ask you to draw. The only thing that came to my mind after last class is their antenna. Read on their antenna. I think they have club-like shape antenna. Club-like shape antenna. You can also check, read on their antenna also. Okay?
Okay, ma'am.
Yeah. So, any other question again?
No, ma.
All right. I am 30 minutes, almost 30 minutes behind schedule. So, what I taught you today, that is the feather, the pineapple, and the orange. You will get your notes today. The rest, I will drop it by weekend. Okay?
Okay, ma.
All
Okay,
right.
ma. Thank you very much, ma.
Thank you, everyone. All right. I wish you best of luck. Please take your time and give yourself the best in your exam. Okay? I wish you all outstanding success in Jesus' name.
Amen.
All right.
Amen.
Thank you. Could they
Thank you, ma.
hack the effect of litmus paper? Litmus, litmus. Maybe no. Maybe no. Maybe they may. But do we have anything that is acidic? Egg is not acidic. Water too is just a neutral water, so it may not have any effect on litmus. They may not have any effect on litmus. Okay. All right. Thank you, everyone. Have a wonderful night rest. And I wish you all best of luck once again. Bye.